Before we start the episode, a quick note. BioBlueprint was created with a simple goal: to bring together the leaders shaping the future of CDMOs and CROs and capture the conversations that really move our industry forward. Every episode is about sharing real insight, real experiences, and lessons that the next generation of leaders can learn from. If you enjoy these conversations and want to see more of them, the best way you can support the podcast is by following and subscribing. It helps us keep growing this community and continue bringing you thoughtful, value-driven discussions with people building the future of our industry. Thanks for being a part of it. And now let's get to the episode. Alex, thank you so much for coming on the show. We'll get right into it. For people who haven't come across Peregrion, in simple terms, what does your company do?
So we help out drug developers by developing faster, cheaper, better. And basically what we do is we use a methodology, ultra sensitive. It's called accelerated mass spectrometer, AMS in short. And this delivers the solution for early information on your drug development.
Fantastic. We'll break down the details later on in the show. So thank you for explaining that in really simple terms. Before we get into the technology itself, let's talk a little bit about what's fundamentally broken in how drugs are developed in today's world.
So I'm not sure it's broken, but what you can see is that the drug discovery at first is highly complex, and only approximately 25% of the drugs reach actually the clinic. Then you start in the clinical trial phase 1, 2, 3, and then you hopefully reach the market. But before you reach the market, already 90% of those drugs did not make it. So from this number, 10% reaches the market. And it could cost up to 1 billion per drug to do this with this limited chance of success. And it could take 20 years to reach your goal. So that's enormous. Yeah, is it broke? Well, I think it could be done better. So there's a lot to gain to get the medicines faster to the patient. It's also saving lives, of course. And that's why.
Okay, so that's really interesting. It's not necessarily things are broken or the system is broken. It could be done better, like you say. The cost of a drug is $1 billion. It can take up to 20 years in terms of getting that drug to the patients. So it's a really long time, and only about 10% of the drugs that are being made actually reach patients. But it could be done in a better and faster way. Am I right in understanding that?
Yeah, you're totally right. That's the summary of it. Yeah, these timelines and these costs, and it's a complex industry. So but it improves each year with new methodologies and discoveries.
So when we think about COVID and the vaccine and how that was developed and how all of that came around, you have people saying the research inside the COVID vaccine was already taking place before COVID existed. Then you have people saying COVID came around, obviously everyone got together and put a lot of effort. That's why we were able to get this vaccine made a lot quicker. So since then, has drug development shifted because of the impact COVID has had?
Yeah, when the COVID vaccine of AstraZeneca was, indeed, there was already a discovery by Oxford University at the time. So was already work done, and then the COVID pandemic broke out. And indeed, there was a lot of focus and concentration and goodwill and of many authorities and people to get these vaccines really quick to the market. So everything was focused only on this particular topic. But then again, the main activity in these 2 years of the COVID pandemic was basically only this, and all the effort was focused on this. After the COVID period, it went back to normal, so to say. But then we also need to realize that there are many drugs to be developed. So the energy is not focused anymore on COVID vaccines only, and that's also the reason why it slows down again. But for sure, the necessity of the COVID vaccines indeed speeded up everything.
I don't know if hype is the right word, but the effort that a lot of countries, a lot of people put together, and the way it was all done and delivered. On one hand, it's amazing, but like you say, it's not a standard just yet. So it will be interesting to see how things pan out in the next 10 to 15 years. But let's go back to what you said earlier. Obviously, these big timelines have
Mhm.
an effect. So why is time such a critical factor in drug development? And what happens if things go wrong much later in the process?
Yeah, that's interesting. At time, of course, you know the saying, time is money. But first of all,
Yeah.
yeah, the longer it takes to develop a drug and to get it to the market, the longer the patient have to wait. And there are
Yeah.
patients, okay, you can save lives, you can improve quality of life. So for this reason, time is important. And secondly, if you can reduce timelines, of course, this saves cost. And shorter timelines also means basically you have a longer patent life for this drug, and you can sell earlier on the drug to the market. So it's, I think, from both ends, patient uh, interests and industry interests. If you have problems late stage in your drug development or in a clinical stage, well, basically what you need to do is to go back to square one and
Mhm.
redo it or repair it or cancel the whole drug altogether. That is the reason why a late stage discovery of a problem in a drug is really wanted.
It's part of the process, and it happens. So just so I understand correctly, and for the audiences here, with with things getting done faster, first and foremost, patients have access to life changing and life saving drugs a lot quicker. But if drugs are made quicker as well, I heard something about a patent. So you're able to hold that patent for a lot longer. A company is able to hold that a lot longer. So those are two things. And then if things were to go wrong in the later stages of a process, then you got to go back to first stage, which
Yeah,
obviously is consuming. So is that right?
that's correct. That in a few sentences, it's all simple, but of course, the whole trajectory and the drug development is so complex with all kinds of in and outs. But basically, that's what's happening, and that's also a reason for speeding up and avoiding unexpected uh, things during this clinical trial procedures.
It's a really interesting point you make, because I was having this conversation with somebody the other day, and some skeptics say, pharma is evil, and so it's not necessarily evil. It's build. It's a business at the end of the day, and it's got to make money in order for it to carry on. Serving patients, revenue in. It's a really important thing. So with the patent point, the fact that obviously you have a longer shelf life on it is good for business at the end of the day. Mostly important is the patient, right?
Yeah, I think that's also true. I mean, I know the stories about big pharma and pharma companies in general, and for sure there must be something true about it. It's huge. The whole industry is huge. Uh, but then again, who else is going to do it? We want to have medicines to be cured.
So true.
So it goes hand in hand.
So
Yeah.
true. So true. Let's move on to drug metabolism and safety testing. So can you walk us through, again, in simple terms, how drug metabolism and safety testing is traditionally done today?
Yeah. So what you can see is the clinical trial stages. You have one, two, three, and then goes to the market. This is really a simple version of complex procedures, I must say. But what is done first before the phase clinical trial one, a preclinical stage is being performed, where animals are being used to test toxicity and also metabolism to find out what could be the effect in humans. So this is the preclinical stage. In stage or phase one clinical trial, a small number of volunteers uh, are being tested to find out whether it's tolerable and also what the dose could be for this particular drug in humans. The next stage is then in a larger group, phase two clinical trial, where also drug-drug interaction, so patients usually have more than one drug, and you want to know what the interaction is. That's a particular study. But also renal impairment studies are being done. So that could mean that a drug have effect on your organs. It's being studied for. And also animal dosimetry studies done. So to find out which metabolites are accumulating in which organ. So that's basically in phase 2. In phase 3, the largest group, because in the meantime we have shown that it's rather safe. It is, the efficacy is there. That's not something when it comes to safety or metabolism studies. And you can take the last leap. And in the last leap, there are many studies, but one of them is also the liquid scintillation counting methodology, where you could also say high doses of radioactivity is being labeled in the drug and then measured back what happened in the body during the metabolism in your body. And that's the late stage. And that's what's being done. In a nutshell, if you take a drug, it will break down in parts. You get metabolites, and those metabolites, that's normal process, could also be toxic or could have another effect. So you want to know for sure what happens with the drug in the human body. And you also want to know basically how long does it stay in your body, in which routing does it leave your body, and what is the concentration actually of those metabolites. And for sure, you also want to know what the metabolites are, because you have known and unknown metabolites. And unknowns are, of course, a no-go. So you need to know what it is. So this is the traditional way of doing, where parts of these studies are being performed in phase 3 with the liquid scintillation counting, which is totally accepted methodology. But then again, in a high dose, and you're not going to apply a high dose to a patient in radioactivity if it's not sure that it reaches the market. Otherwise, it will be unethical, I would say. That's the traditional way how it's being done today. And there are many variants and other forms of it, but that's a bit too far to go into that.
I like what you say when you talk about it in simple terms in a few sentences. It's easy, but when you actually break down, preclinical stage, where animals are involved, the metabolism side of things, and then every step after that, it is a really complicated thing to play out. It's
Indeed.
simple as said, as you say. And so, if I was again, what I want to understand is when people are watching, not just necessarily in the life sciences space, but maybe outside of that, it's a really simple thing to understand. So when you get to the point where you mentioned the liquid scintillation counting, that is when you take a drug, how it affects in your body, how it breaks down in your body. That's really what that whole thing is. And you gotta, if you have a certain target you're aiming for in the body, you got to know how it's playing out inside a person's biology and body. Am I
Indeed.
right in saying?
Yeah, I think that's a good summary. Yeah,
Okay.
let's hope
Okay.
for it. Yeah.
Okay. So then what's, when you look at your company,
Mm-hmm.
mentioned AMS technology earlier, the microtracer AMS technology.
Yeah.
How things are done traditionally is what you just explained, and the efficacy and all of that is attached to it. So let's talk about the solution you guys are providing. What exactly is microtracer AMS technology?
Yeah. So the microtrace technology is actually a methodology to find those metabolites really, really early in the clinical stage. AMS technology is already used in the industry, but still in a late stage, 2B. The next phase would be 3. So that's quite okay. And it's a little bit competitive currently still with the liquid scintillation counting. But the methodology as such, the microtracing, because of the sensitivity and the really low dose of radioactivity, you can apply this already in phase 1. And that seems to be only shifting to numbers, but it has a huge implication. Because actually in phase 1, you already can understand, measure, see what happens with these drugs in humans actually. When it breaks down in metabolites, you can also detect what it is and if there's any unknown metabolite coming up. Except for this early stage, it's also really a low dose. That's totally ethical in all respects. And you can even say that it's totally safe for all men, including children, women. And that's also a game changer, I think, pharmaceutical industry. There are many considerations to test new drugs in women and children next to this story. So the sensitivity of AMS, accelerated mass spectrometer, that's the game changer. And then we're talking about 250 to 2,000 times more sensitive than the scintillation counting. And because of the sensitivity, you can do much more for your drug development and drug discovery and also in the clinical trials. It brings a lot because if you think about it, first of all, certain aspects during these clinical trials from phase 1 to 3, you can skip. The number of animal testing, you can skip. You can do it early and you will discover unknown metabolites already ahead of the race. You don't have to go back to phase 1 or square one if you discover metabolites in phase 3, which is the current more or less situation. Those upsides are huge. And that's what we do. We have developed in the recent 8 years, together with TNO, it's a Dutch applied sciences organization, an AMS system. It's being driven by Wouter Vaas, our CSO. And we were the first to have the system up and running and perform this for the pharmaceutical industry. In the meantime, we work with big names in the industry, the big pharma companies, but also CROs. And it becomes more and more implemented. Basically, what we're looking for, the paradigm change, because we have the existing methodologies, as we discussed, but there is much to win to implement it and to bring it to phase 1. And the upsides we discussed already, but to get from 2B or phase 3 into phase 1 with AMS technology, that's not a given in the pharmaceutical industry. And there are many reasons for this, of course. First of all, it's a huge, fast industry with huge organizations. It's, I think, risk averse, and I think that also serves a goal because you don't want to have any medicines which are unsafe in any respects for people, for patients. So that's quite okay. But it's also holding up a little bit the advancing to phase 1. So that's what we do with microtracing technology. And it's working out really well.
So loads to unpack there. And from my understanding and from what I'm hearing, obviously there's still a lot of work to be done across the industry and all of that. But there are clear benefits that this technology is providing. Like you say, AMS is being used in the industry already, and it has been used, but in the later stages. With how you guys are developing this, you can use it much earlier, which negates the potential of using animals in the study. Then, in terms of the ethical side of things, you mentioned obviously for males, females, children, it's a lot more safer, which makes it then ethical. And then obviously your CSO you mentioned is developing this. So a lot is happening, and you're working with some big pharma already. Is that right?
That's correct. Yeah. So big pharma companies recognize the value, the added value for clinical trials. So they use it. So we deliver this service to them. And of course, the biggest advantage is if you use it earlier. And this step to go earlier, that's of course the whole discussion here. What is holding up the progress to phase 1? And that's what we also try to communicate into the market, to regulators, to our customers, to other interest groups, to understand that this would be the way to use AMS. Yeah, it's also being seen by regulators like FDA, NIH. First of all, they have issued guidelines in 2024 where they ask and recommend to have metabolic information data as soon as possible in the whole clinical stage. So it actually supports, it's a tailwind to use the methodology in phase 1 and not late stage. Furthermore, there's also a draft guideline this year from the FDA, NIH, to abandon animal studies. It's complex, but over time this will happen. But at this moment in time, already AMS technology supports this wish. So that's also a reason and tailwind for us to motivate to use AMS early on.
How far are we from a place where we live in a world where animal testing is completely out? Do you think it's going to happen in the next 15 years, 10 years, 20 years?
I think some time away because, it's a trade-off. Do you use animals and do you have not all information you need for safety purposes? And do you take risk with your patients? Well, there's no question any authority will not accept that. But it's also about new methodologies being developed, in vitro methodologies, so outside the human body. But also AI modeling is really speeding up. So I think that this moment in time will be also depending on those new methodologies and techniques, and then it will go out perhaps within 5 to 10 years. But sooner, I don't expect.
Because things have to go hand in hand. While you're developing new methodologies and while these things are in play, there isn't a clear-cut solution right now that exists. And because it's such a big thing, it will take some time. Earlier, you mentioned lower levels of radioactivity. Why is this so important? When we talk about radioactivity, how low are we talking, and why does this thing in particular matter?
Radiation is not healthy for a human being in general. Okay, there are therapies to cure, but that's for a good reason, of course. But in general, it's not healthy. So the less radiation you use or exposed to is better. So if you use a high radiation level in your clinical trial development or in clinical stages, phase 3, that is, unwanted. If it's necessary, it will be used. It will be tested by medical ethical committees. The lower the radiation level is in the tested drug, the better it is. And we use basically 100 plus fold lower radiation levels than the current methodology, liquid scintillation counting. And that's enormous upside, of course. And that's also the reason why it's accepted and allowed to use this already in phase 1 and not the high dose. And we're talking about in the range of 1 microcurie. That's a unit for radiation. And that's actually really low. So if you take a plane and you fly from Amsterdam to London, then you already have being exposed to 1 microcurie already. And that is because you're higher up and cosmic radiation causes this actually. So it's important. And there also you have the ethical topic, because you can use lower radiation levels. So if you can, please do it.
So radiation is bad for humans. We all know that. And basically, the idea behind this whole thing is that you're using a significantly less levels of radiation. I think you mentioned 100-fold lower than
Indeed.
liquid scintillation counting.
Yeah.
And obviously, that is just a better thing for everyone involved. Is that
Indeed.
in simple terms? Would that be right to say?
I think so. Yeah, it's right. So the sensitivity of the AMS allows you to use 100-fold lower radiation in the trial. So it goes hand in hand. It's just better, I would say.
Amazing.
Yeah.
Amazing. And obviously, the fact that you're getting recognition from big pharma and you're working with big pharma, there really is a strong case for this to exist in the world. So I'm gonna move on the conversation here. We've spoken about the pros, the cons when it comes to the current things we have in place with AMS, what you guys are developing with this technology and all of that. Um, just I think we may have covered this already, but just from a scientific standpoint, what makes AMS more accurate than the methods we have available today, like liquid scintillation?
So basically, the device we use, the accelerated mass spectrometer, is so much sensitive by the way how it has been built and how it's been working, that this is the cornerstone of the whole methodology and the reason why we can do all this. In summary, so much less radiation of the label in the tested drug, and so much earlier in the clinical trials. That's all because of this sensitivity of the device.
Understood. Understood. And we've highlighted the benefits for this already. So
Mhm.
in terms of time saving, this is another important part in drug development. As we said at the start, it takes so long to get a drug to a patient. The amount of money that's invested in developing a drug, it's so much. It's such a big puzzle, but time is of the essence and so critical. So you mentioned companies can save up to 2 years when it comes to the timing of all of this. How significant is that when it comes to the cost and competitive advantage for companies?
So if you use uh, metabolic testing early on phase one, um, you take also the benefits which goes along. It saves standard by standard approximately 10 months already. So you're 10 months earlier on the market. You can sell the product 10 months earlier, and your patent life also will last longer. So that's protected for copying. So that's important case. Uh, what is even more important is you should avoid um, the discovery of the unknown. Go back to phase one after you have reviewed and repaired the drug. So go back, that means again through all those stages. That costs years and millions to do so. So it's, and that could last years indeed. And so the benefit is you de-risk actually by testing metabolism in phase one. And de-risking is important because you avoid potential risks. By definition, you are using this method 10 months earlier. And it's also interesting for big pharma, but also for smaller companies, biotech companies using and developing small molecules, because any investor wants to know, is this drug actually a valuable drug? Are we looking forward to exposed to risks in a later stage of the clinical trials? So it's also an upside for them to present to investors. And we have here a drug. It already passed discovery. We're in phase one or phase two. And yes, we have done all the metabolite studies. We are in a green zone. We are safe. That's a really good pitch for smaller companies to present to potential investors. For more purposes or more types of companies interesting to review this and see this, take this along in their business case.
So if I'm running a small business and a small company, using this technology, uh, the the pitch that we can put forward to a potential investor, uh, as far as I understand, becomes a lot more credible and a lot easier because you have all of this evidence. You're mitigating risk, like you said, de-risking avoids potential risks, and then when you present that to a potential investor, it makes the conversation a lot easier. So, if I'm a investor and I have all of this evidence for a small company, does investing then become easier because of all of this?
I would say yes. And of course, that's the easy answer on a very complex question. I mean, there's so many uh, factors which makes a investor go into it. It has to do with the indication, what the other data tells them um, about the team of such a biotech company, et cetera, et cetera. So that's endless. They have their ways to evaluate that, of course. But at least you can say we know a lot about our new drug,
Yeah.
and we have de-risked the whole thing because we have all the understanding about our the metabolites because we tested this already in phase 1. So there's really a big upside.
Yeah. Okay, fine. Are de-risking, and so when you talk about de-risking in this context, is that the same as helping to basically reduce the risk of failure in drug development? Is that the same thing?
Yes, it is. I mean, there are, I think, more than 100 reasons why a drug will fail in a clinical trial. It's not only about the metabolites. It could not be efficacious, or it could be something else, another risk, a problem, or I don't know.
Yeah.
It's hardcore de-risking.
Okay. So then, if the benefits are so clear and the advantages are so clear, um, I know pharma and life sciences is such a big, big thing. And to bring that into standard practice will take time. But I'm going to ask you the question anyway. Why hasn't the industry already adopted to this?
So they did adopt it, but used still in a late stage. They recognize, acknowledge also the technology, but to make the next step to the phase 1 early on, that's a different thing. And like we discussed, it has to do with the complexity of drug development, which is not only metabolism. It's also about huge organizations with different departments, with their own interests, professional interests, deliberations. So it is a machinery which needs to gradually understand and accept that there are better ways. And I'm only speaking now for what we do and metabolite studies, including all the other services. And of course, in this whole drug development, there are so many more factors to evaluate. So, of course, I would like to stand up and say, listen, there's only one thing important, and that's AMS technology, because we are here. No, it's much, much more. And therefore, the change or paradigm change is not really going fast. But if you add up all things, so the FDA and NIH are in favor, they want to have it, less animal studies. You have so much more intel upfront, et cetera, et cetera. So it's a no-brainer, but it needs to be integrated step by step. It will take time, also in the larger corporate companies. Well, you also have the smaller companies. Sometimes they are short in time. They need to make a run for phase 1 or phase 2 trial. Don't want to be burdened with new complexity. So, but it could also be that we have not communicated properly to everybody what it entails and why it's so important. So there's also a part with us where we should, express ourselves a little bit more in the forefront on a global scale.
Um, for your technology, would you say you guys are the pioneers in applying this to the pharma space?
Yes, we are the pioneers, and I can also say our Dr. Vaas, CSO of the company, already 8 years back, he started to develop this methodology. He was the first. We brought it to the market with the spin-off Peregrion, because we're a spin-off of the TNO organization here. So we were the only one with AMS in the market. And in the meantime, you can imagine it has been copied, and there are more providers. I see this as a compliment, actually, because apparently it makes sense to do it, and this is being recognized by pharma and also other providers. So it also generates competition in our market, and I think that's proof that there's need for those technologies and these services. And I'm more good in that respect.
it's not often people see competition as a good thing. You're seeing it as, if companies are obviously copying, it's a compliment to you guys. Like you say, 8 years ago, your CSO developed this and he's worked on this, and now you're seeing other companies come up with a similar thing or maybe the same thing. But that's a good sign, because it means more people are trying to make this happen. You know
Indeed.
what I mean?
Yeah, for sure. And that's precisely what it is. So it's a big world, and there are many drugs being developed. So um, if the AMS technology is being used more frequently in the future because it gears up to phase one, there are more drugs than in this stage. The total capacity needed is also higher. So yeah, it's what it is, and I don't consider this as a negative aspect.
Amazing.
Competition, yeah,
That's an amazing view to have. Um,
yeah.
how do you position yourself in the market then? So, are you a tech company? Are you a service provider, or
Mhm.
something entirely different?
Yeah, what we actually do, we bring, we solve problems for complex drug development programs. That's what we do. So we have a great team over here, a PhD team with a lot of experience in clinical trial design. What we do is we go to drug developers in big pharma, smaller companies. We want to be at the table as soon as possible, as early as possible, to explain, to help out for the design of the study. Um, um, that's the first thing we do, and basically also the most important thing we do, because the AMS technology, using the technology as such, that's our core activity. That's measuring, of course, but bringing the solution, that's basically what we do. And yeah, you could call it a service, but that's so common, you know.
Okay. Okay. Understood. Understood. Let's fast forward 5 years, right? If you were to have this conversation in 5 years, what do you think will have changed in drug development?
First of all, but that can't be differently. The LSC, liquid scintillation counting, will be fading out. That's what I expect, and the AMS technology will be the only methodology. But in those 5 years, I also expect that a lot of new methodologies are being developed when it comes to um, uh, uh, testing for clinical trial for metabolites, but also, as we discussed, the AI modeling is coming up. So I think that the total picture in 5 years will be totally different, likely quicker, more efficient. But then again, you have to call me in 2030 because I'm not really 100% sure. Yeah.
Yeah, yeah, yeah. So LSC will be fading out. AMS obviously gaining more traction, and will be the hopefully the more dominant technology in the market.
Hmm.
And that AI modeling, like you say, all of that is basically going to enable things to happen a lot more quicker and hopefully more efficiently. But you don't know what the future holds. That's the hope, and that's the idea. Um, so I want to ask you one last question from my side. What's what's your message to pharma or biotech companies?
My message would be smart, test your metabolites as early as possible in phase one. Ethical components, de-risking, saving timelines, saving money, sooner drugs for patients. That's it.
And that's a strong message. And we'll end it on that. Thank you so much for coming on the show. We wish you and the whole company continued success, continued growth. And in 2030, when we have this conversation again, hopefully it's as we hope. So thank you so much.
Thank you, Leonard, for being in the podcast. It was really a pleasure, and hope to see you soon. Bye.